Hello, and thank you for your interest in our poster. My presentation is titled Genome-scale Modeling of Pseudomonas aeruginosa P14 Unveils Broad Metabolic Capabilities and Role of Metabolism in Virulence and Drug Potentiation. This work is currently available on bioRxiv. Pseudomonas aeruginosa is one of the leading causes of nosocomial infections. Here with this pie chart, we can see that Pseudomonas is prevalent in hospital wards with highest prevalence in intensive care units. Once transmitted, the pathogen first colonizes the host before establishing infection, and at chronic infection, biofilm production occurs. Once the infection is established, it is difficult to eradicate, and one of the primary reasons for this issue is the presence of multiple mechanisms of drug resistance, which are outlined here. For instance, overexpression of efflux pumps, lipid modification, etc. This is the overall method we have utilized in our study. First, we reconstruct the metabolic network of an organism, and this network is decomposed into stoichiometric matrix, such as that the reactions are columns and metabolites are the rows. This matrix can then be used or can be studied using mathematical optimization approach such as constraint-based modeling. We used a semi-automated approach in order to develop the model. First, CarMe was utilized to develop a draft reconstruction. Then onto this reconstruction, we added the biomass reaction from an already existing model of PA14. Then we performed simulations and gap filling until the model was able to simulate growth in rich media condition. Following this, the model was iteratively extended by filling in missing gaps and quality control to finally get to the V4 draft model. Then, organism-specific reactions related to phenazine-dependent metabolism, carnitine metabolism were added to the model to finally get to the model IST1507. We assessed the model for its ability to predict gene essentiality and substrate utilization. For gene essentiality, we performed simulations in LB-rich media condition, synthetic cystic fibrosis media, and glucose minimal media. And we optimized the model for biomass production. As can be seen here, the model is able to predict gene essentiality in all three different media conditions with an accuracy of greater than 90%. Next, 123 substrates in minimal media condition were tested for substrate utilization, and biomass production was used as the objective. As can be seen here, the model is able to predict substrate utilization with an accuracy of 93.5%. An interesting case study using the model is related to phenazines. Phenazines are cofactors that can be reduced within the cell, and then they are transported out to the extracellular where they get oxidized, and the oxidized phenazines can be transported back to the cell. To account for the growth associated dilution of cofactors such as phenazines, we used a limed FP approach. Then using the limed FP model, we performed our simulations. To simulate oxygen availability to the cell, we chose to constrain oxygen import flux from extracellular to the periplasm. Then we determined the effect of piocyanin production on growth rate at various oxygen availabilities. The simulations suggest that under low oxygen condition, as seen here at the bottom, with increasing piocyanin production, biomass production also improves linearly. This, however, is not true as more oxygen becomes available, as can be observed in the top section of the map. This all indicates that piocyanin production is important for the survival of Pseudomonas in oxygen-limited conditions, such as those encountered within biofilms. Next, we use the model to study gluconate production in different mutants. Out of 13 mutants, we found that the deletion of EDD gene, which encodes for 6-phosphogluconate dehydratase, lead to higher yield of gluconate compared to the wild type. In the heat map, the values are obtained by dividing the fluxes or yield of the mutant by the respective yield fluxes or yield of the wild type. Then we used flux variability analysis to mechanistically explain the gluconate overproduction phenotype of the EDT mutant. In the graph in the figure, the points refer to the maximum and minimum fluxes of the specific reactions, and the dotted lines refer to the flux range. The values right after the reaction names are derived by dividing the average flux of the mutant by that of the wild type. We see that the gluconate secretion is higher in the mutant than in wild type, with only one flux value. This all suggests that for the mutant to grow optimally, it must secrete gluconate at the expense of biomass production, which is considerably lower than that of the wild type, as can be seen here in the heat map. Finally, we used the model to study tobramycin potentiation by fumarate and tobramycin protection by glyoxylate. The experimental study was published in 2017 by Malin et al. We performed flux sampling analysis which samples all the feasible flux solutions for a given condition. As can be seen here, when the fluxes were compared between the two conditions, the TCA cycle flux overall is higher in fumarate, as can be seen through the blue arrows. Furthermore, the oxygen uptake rate and proton motive force is also higher in fumarate. Likewise, in glyoxylate, the flux is diverted from TCA cycle towards the reaction catalyzed by malate synthase, which leads to lower TCA cycle flux. These simulations hence recapture the results of Malin's study that demonstrated that upregulation of proton flux and TCA cycle leads to increased tobramycin uptake rate and activity in fumarate supplemented media, whereas shunting of the flux by glyoxylate lead to lower respiration rate and reduced drug lethality. Therefore, using the constraint-based modeling approach, the effect of metabolism on drug uptake and potentiation can be investigated. And that concludes my presentation. I would like to thank everyone involved in this project, especially my advisor, Dr. Lawrence Yang, who has been tremendously helpful, patient, and considerate.